I'd like to thank the Craft COVID Committee on behalf of our team for reviewing our proposal, and I thank the VCR's office for the generous funding to try and develop a new mouse model to investigate coronavirus infection and pathology. So this is a joint multi-PI project with myself. My role in this really is design and utility of our existing TMF, transgenic mouse facility on campus, which is a bit of a, I think, a jewel in the crown at UCI, to generate a new mouse model that can be used to study coronavirus infection. And Kim Green, Tom Lane, and Eric Perlman are the folks that are really going to do the difficult work in terms of analyzing a new model and determining the utility of this. All right, so the project overview for this committee, you don't need to look at all this. I'm sure you're all acutely aware of the issues with regards to SARS-CoV-2. The only points I'd like to make here are the bottom bullet that SARS and SARS-CoV-2 both use the human angiotensin converting enzyme 2 transmembrane protein as a receptor for infection. And clearly there's a need for small animal models that will reproduce a clinical course and pathology as found in COVID patients. The statement at the top here, this is something that many people are often acutely critical of. That is, the mouse is not a human and vice versa. The statement or the quote there from Judah Folkman many years ago, this was in response to a quip from Jim Watson that Judah was going to cure cancer in 2 years. The bottom line here is that I think in the past 20 years or so, excuse me, because largely of the advent of good computers, high computing power, high throughput, omics type strategies, we're now much better positioned to be able to convert findings in animal models to findings in humans and vice versa. And so I'm much less pessimistic now about the challenges. They still remain, but I think that the mouse is and continues to be and will improve as a valuable model system to understand pathology in human genetic disease as well as infectious disease. So one of the key challenges here, of course, is that wild-type mice are resistant to SARS and SARS-CoV-2. Some of the reasons for that are shown on this slide. There are a variety of polymorphisms between the protease domain and humanase 2, that's shown here in blue, and the SARS-CoV-2 receptor binding domain shown here in gold. And these polymorphisms are probably responsible, along potentially with some glycosylation changes, for the inability to infect mice effectively with SARS and SARS-CoV-2. So this is a challenge, and of course we can overcome this by doing site-directed mutagenesis, etc. But there are other challenges in using the mouse as a model system. And one that's been appreciated relatively recently is that the human angiotensin converting enzyme 2, or ACE2 gene, is actually an interferon inducible gene. So this is a little bit sinister because, or serendipitous if you're the virus, because often with viral infections, you will induce interferon production. So this can lead to a feedback mechanism, a positive feedback mechanism, where viral infection and the body's response, ironically, makes the body more susceptible to repeated infection. On the left here is just shown inducibility of interferon, of ACE2 in response to interferons, both type 1 and type 2. And on the right, potentially the reasons for this, on the upper part of the diagram here, I don't know if you can see my annotation with humans. You can see here that there are both different STAT2 elements and in interferon response factor binding sites here between human and mouse that may be responsible for these differences in responsiveness. So what about existing mouse models? Why should we bother making a new one? Well, the existing mouse models are somewhat limited. At the top here, I haven't listed all of them, just the ones that are popular and are recent. At the top, you can see here a keratinase 2 model, which is very popular. This has pros and cons. The pros are it expresses ACE2 at very high levels in many epithelia, importantly including the bronchial airways. So this is actually a useful model if you want to look at the acute response, including things such as inflammatory response and infiltration of a variety of cell types. However, one of the drawbacks of this is you do have supraphysiological levels of expression of ACE2 and the lack of expression of ACE2 in all of the clinically relevant cell types. So alveolar type 2 cells that secrete a surfactant are not infected here, at least not initially. Also, this model is not inducible by IFN, so crude but an effective model. The human ACE2 mouse, there was one recently published by a group in China. They knocked in the human ACE2 cDNA into the mouse ACE2 locus, and they also included a fluorescent reporter gene to track the cells expressing this. So this is a nice model, but again, because mouse ACE2 is not induced by type 1 and type 2 interferons, this has a little bit limited utility. Also, that model is not going to be available in the US for a while, and we have no idea about the MTAs that may be restrictive there. Lastly, more recently, another crude but effective method is to use replication-deficient adenoviral infection. This gives you great levels of ACE2 in a variety of cell types, but again, supraphysiological expression of ACE2, variability in ACE2 expression, and there's no extrapulmonary disease in that system. Okay, so how are we going to try and build a better mouse? Two aims here. One is to make a mouse model that expresses physiological levels of ACE2 under regulatory control of the human ACE2 locus and doesn't make its own mouse ACE2. And then the second part that Kim and Tom and Eric's labs will do is to demonstrate proof of principle and compare this model with the K18 ACE2 model. How are we going to do this? Well, the first part, and I'm being deliberately a little bit light on the details here. I'm happy to share details offline with people if you're interested. The first part here really involves identifying a part of the human genome, and these are available in what are called BAC clones or bacterial artificial chromosome clones that contain ACE2 and the required regulatory elements to mediate the proper expression of this in the appropriate cell types. And so we can engineer this. We're also going to try and do this via targeted transgenesis to insert this into the mouse genome. And we'll then screen the lines initially for expression of ACE2, including interferon inducibility. The second aim here, sorry, it seems to be locking up for some reason here. There we go. The second aim here then is to actually mutate the mouse ACE2 gene. This is relatively easy to do, relatively straightforward. We'll target this with CRISPR-Cas9 and mutate this so the mouse no longer makes any Cas9. No longer makes any ACE2. And then in aim 2, once we have bred those animals together, validated expression of human ACE2, we can then do the actual infectious work where we can take the new humanized ACE2 back, ACE2 null mouse model, and subject that to infection with SARS-CoV-2 isolates, initially by intranasal inoculation, and then look at survival, viral load, look at the histopathology, including what happens with interferon treatments, perhaps, and look at the immune cell infiltrates in these models, and compare the utility of this new model to the previous model, and also obviously with control wild-type mice. In terms of impact, we think that this will be an improved model that may be more physiologically relevant. We think it'll really inform about the immunobiology and pathological mechanisms of SARS-CoV-2 infection. Plus, we can then use the power of mouse genetics, including a variety of different knock-ins and knockouts that are already made, and combine these with this model. And we can also cross this model onto genetically diverse mice to look for risk loci that may play a role in the human condition and inform as to additional risk factors involved with SARS-CoV-2 infection and or outcomes. So I'll stop there and would be glad to answer any questions. I'd also mention that Eric and Kim are on the call as well, so I'm sure they'll be happy to participate.
So this is Eric. Grant, that was nicely presented there. But maybe I missed it, but did you mention the timeline for generating these mice?
No, I didn't mention a timeline, Eric, sorry. So the production of the ACE2 knockout mice should be relatively quick, but what will take a bit of time is engineering the backs and validating the humanized back animals. So I would envisage that this model, ready to go in terms of being infected, it might take at least a year. So obviously one of the key challenges for investigators is, well, what mouse model do you use just now? Frankly, I would suggest that despite its limitations, the K18 ACE2 mouse is probably the best one to use. As I mentioned, you can study acute infection and look at cellular infiltrates and acute responses. And while it may not be the best, it's going to be the best thing for now.
So Grant, I have a question along those same lines, I guess. Given that there are questions about how well this would recapitulate the activity of the human version, I guess, is it faster to do some replacement experiments with the human promoter in mouse cells just to see what the responsiveness is, whether the promoter is comparable in activity, and have those experiments been done?
Yeah, good idea, Suzanne. Unfortunately, those are what I would call high risk, high reward. There's more to life than a promoter, and despite our knowledge of differences in interferon response elements, etc., it's a bit capricious to go putting a human promoter into the mouse locus. Plus, given the codon sequences changes that probably preclude infection of the mouse in the protease domain of ACE2, you'd have to actually CRISPR multiple regions of the mouse ACE2 coding sequences. So that would take at least 2 rounds of CRISPRing, which might take up to 6 months. So, you know, we thought through these issues pretty long and hard, and I think the approach that we're using, given the resources available, will be a pretty solid one.
I guess to come back in a different way, how did you go about defining the extent of the human promoter?
Yeah, so without going into all the details, we use a variety of algorithms to look, comparative genomics to look at the human ACE2 locus. We look at non-coding transcripts that may be expressed nearby that locus, for example, antisense transcripts that may play a role in regulating expression. We look at response elements, we look at conserved sequences, and based upon that, we make a decision on where to trim our backs in order to hopefully get all the bits that we need. Looking at TAD domains, or TADs rather, is also important. So we know that the ACE2 sits within an individual TAD, so we don't have to worry about spanning TAD boundaries.
Okay, okay, thank you.